I just wanted to follow up on the point that Alec Markham made just before lunch, which was that we are at a tipping point. And I think he's dead right. I haven't got time to go into it now, but basically the next stage of the argument, which I'm going to be presenting here, is concerned with, would be concerned with the financing of biobanks in a larger context. And when you think about how financing of research more broadly is organized in this country, that would lead you to suggest that there may be improvements that could be made to that. And now is probably a good time to be thinking about new ways of funding research as a whole. But here today, we're just going to be talking about the financing of biobanks with a rationale for a national biobanking research infrastructure. And the proposal for this comes out of the STRATUM project that Chris Christodoulou mentioned. That project was funded by Innovate UK to look at new business models and value systems. That was the funding stream that Innovate UK funded us through. The sponsors were AstraZeneca, GSK, and AZ produced the initial proposal. The aim was essentially to specify methods to increase the effectiveness of sample provision in the UK, and that aim was endorsed by those organisations, and the consortium that we put together comprised the public sector, the private sector, the third sector Genetic Alliance, and the PI was the redoubtable Julie Caulfield. You can see all the documents that were produced in the project at that website there, and that I think would give you a very good a proposal for the construction of a national biobanking network overall. We were concerned with one of the work packages. There's all the other ones listed down there, which was the business, which was the business model. And so our task was to review business models and to identify a sustainable one. The investigators were at the University of Manchester from the Institute of Innovation Research there, Sally G and Luke Georgiou, who's also our Vice President for Research and Innovation, and from Sigma, which was set up by Bill Ollier, were Rob and myself as one of the co-directors of Sigma. The method that Sally and Luke had decided to adopt was the case study, because it enables in-depth examination and various other reasons. And the data sources were primary data from semi-structured interviews and secondary data from minutes, press releases, and so on. The thing has been, the paper that arose from the work that we did has been accepted for publication. It should be available later in the year. Okay, the cases studied were these: Abcodia, AstraZeneca itself, Sigma Biobank, Fresh Tissue Supply, Nottingham Health Services Biobank, a small research collection, UK Biobank, and the UK Brain Bank Net. Oh, why are there three Bs? Because I can't type. Brain Bank Network, BBN. British, no, it's just two Bs. and as you can see, the location and ownership was, well, Abcodia is a university spin-out with ownership by venture capital, other private capital, and the university. AstraZeneca is a corporate biobank, separate from the MedImmune biobank, which was what I asked about. Sigma, of course, is university located and owned. Fresh Tissue Supply, hospital and university, university research group and hospital, independent charity for UK Biobank, and university and hospital for the Brain Bank Network. Okay, so one of the tasks that was set was to discover the costs in the first place, because STRATUM had initially believed that discovering biobanking costs would yield a consensus price list, a cost model that the community might accept. So cost and other data were collected via questionnaires, face-to-face meetings and follow-ups. And what we found was that the response rate and the level of detail were variable from bank to bank, and the detailed comparable data were not obtained. So you'd say pretty bad result. However, that itself is a result, because analysis identified the reasons for the lack of detail and comparability. And those boil down to these headings: hidden costs, variable cross-subsidies. Okay, so most costs, many costs, cost items in biobanks tend to be hidden away. You can't find them. And there are variable cross-subsidies between research and from estates and all sorts of different ways in which the true cost can't be established. There are opaque financial systems in the NHS, would you believe, and in universities. there was an inability to provide financial data when it was requested. And that's mainly because biobanks and organizations like universities have no regulatory obligation to provide that level of financial data. And there was also unwillingness to provide financial data for commercial considerations or confidentiality considerations. So, first conclusion was that a consensus price list is unrealistic. However, note that there has been an attempt to develop an algorithm to specify prices in biobanks from a EU-US expert group. And the method worked, but the prices were unrealistic. And you can read about it in Science Translational Medicine, Clement, Yule, and others last year. The second conclusion is that biobanking is, in socioeconomic terms, inefficient and ineffective. And this then causes a negative impact on research and on delivering stratified medicine, and then leads on to a strong rationale for reform in a situation of market failure, which I'm going to come back to. So how do biobanks set their prices? Well, it emerged from the case studies that they calculate prices to cover supply costs. But often when that is sought to be done, they don't include samples in their accrual, storage, or infrastructure. To cover direct costs only, but different banks use different criteria to define what direct costs were, to cover both direct and indirect costs. And the final thing was that respondents said that prices actually reflected the ability or willingness of users to pay. Hence, price levels are not set by the market and are below cost. The price isn't right. This corresponds to market failure. Market failure does not mean that profit generation is always impossible, but there is no evidence of profits in any of the STRATUM cases. And elsewhere, commercial biobanks may exist and they may operate, but at low margins and hence are vulnerable as organizations. And Astrand was one of the examples that we cited of that vulnerability in our report. So the ways in which these different cases are financed are as follows. Nearly all of them are from what we call mixed public funds, so Research Council, project grants from the institution itself. And the only exceptions were AstraZeneca, which of course is corporately funded, and Abcodia, which in addition to mixed public funds also has charitable funding, venture capital funding, and other private funding. So that's the way in which they're financed. And this spectrum of funding corresponds to a survey that was done on US biobanks a few years back, which showed that 78% of banks there are funded by, essentially from mixed public funds. Okay, so market failure and biobanking. Market failure is conventionally defined as when there is no match between supply and demand. Usually there may be a complete absence of supply, in which case there's total market failure, or there's only partial supply, it doesn't match demand. Either condition is considered to constitute market failure, and the contributing factors can be what are called negative externalities or positive externalities, monopolies, impairment or immobility of production inputs, and market information breakdown. Okay, so in biobanking, we concluded that market failure was expressed globally as a combination of two things: lack of coordination between biobanks plus poor visibility of samples, leading to impeded access to sufficient samples. And some of the specific features of market failure that we identified were that the law forbids certain transactions. You can't flog human bits and pieces on eBay. Ethics restricts transactions. I'm not saying it shouldn't restrict transactions. I'm saying it does restrict transactions, whether you like it or not. Annotations are usually not current, so that boils down to market information breakdown, not enough information about what's going on. Inconsistent for quality and ontology, well, that probably corresponds to impairment or immobility of production inputs. Price set by user's funder, I think that's an impairment, another impairment. Inaccessible, that's market information. Poor visibility, market information breakdown. So a number of features of biobanking are reflective of market failure. So what's to be done? Well, first of all, we should take courage from the fact that the bioreagents market and technology platforms, by and large, there is broadly a functioning market in those, and nobody really is worrying about the availability of monoclonal antibodies, for example. But for human biological samples, there is not such a functioning market. Economists of all stripes, even if they use different language, consider that public funding of research is essential. And here are the main reasons that they give: that the output of research has the properties of a public good. A public good has a number of particular characteristics to it. One of them is non-rivalrous consumption. If I buy a bottle of Coke, then it's my bottle of Coke, and that's the end of the matter. It's just mine. So whereas knowledge, if I obtain knowledge, there's absolutely no reason why another person shouldn't obtain exactly the same knowledge. Yes, there's no prevention. That bottle of Coke is mine, and it's going to go down my throat or down the sink. But that is not the case with knowledge. Generating knowledge stimulates the generation of new knowledge, whereas generating Coke doesn't necessarily stimulate the generation of more Coke. Benefits of research are widespread, diffuse, and unpredictable. There's limited access. If there is limited access to knowledge, then this slows the pace of innovation. Research is a long way from market. This disincentivizes firms from investing in R&D. Economists advocate public funding in situations of market failure. So is public funding of biobanking justified because it is part of research and or because there is a market failure? Either way, reform is required. It doesn't really matter which way you look at it. Okay, so can we find a better biobanking business model? And I don't mean that kind of model. Market failure is not the end of the world. The identification of market failure should simply prompt reforms. For example, the Department of Business examined last year 8 types of failure in 24 innovation areas across the UK. Okay, 8 types of failure in 24 innovation areas. There's a lot of innovation areas. And BIS, not DIS, says infrastructure failure is likely to be a showstopper, which was defined in the report as a barrier that effectively makes successful innovation completely impossible or almost completely impossible. So infrastructure failure is a particularly nasty type of failure, and clearly could become a showstopper. And I think in some ways the problem with biobanking is that because we've not resolved the problems, it is a bit of a showstopper. It is really stopping progress, really, really, really. So 2 types of reform are available. Either we can try to fix individual issues one at a time and hope for the best. For example, an effort to address one biobanking issue, that of poor visibility, is being undertaken by Anne Carter and her colleagues via funding from a basket of funders, but only for 3 years. Or we can adopt a better business model. And the distributed research infrastructure model is a potential new model that we could, and I would argue should, adopt, and what STRATUM actually says we should adopt. The UK government has just recently applied for membership of the European Biobanking Network organisation called BBMRI-ERIC. So this is the intergovernmental body that runs the emerging European distributed biobanking network. So this is a very first step towards developing a national network, but it does show that this government, like the previous government, ultimately is prepared to accept that a biobanking network is required. Just to tell you more precisely what a distributed research infrastructure is, research infrastructures are any facilities, resources and related services that are used to support knowledge creation and distribution. And they serve, the key point about them is they serve internal and external users. That's the critical thing. There has to be a degree of access, in other words, because they serve external users as well as internal ones. And of course they're well established in the physical sciences. In fact, we couldn't have had most of the discoveries in big science over the last few years without such research infrastructure. And here's a picture of the Large Hadron Collider. Distributed research infrastructure comprise facilities that are best provided across more than one location. So that's the only distinction. They're provided across more than one location. A good example is the European Bioinformatics Institute's lead on the European research infrastructure in bioinformatics called Elixir, and this provides services for bioinformatics and bioinformaticians across multiple sites all over the European Union. Distributed research infrastructure requires coordination. That's the key difference between it and an ordinary research. I mean, they require coordination within themselves, but these people, but a distributed one clearly requires coordination across all the distributed parts. So the sites require a shared strategy and common standards for governance, training, materials and data. That's why I asked Tim about common standards in relation to UK Biobank and other large epidemiological cohorts elsewhere in Europe in particular. STRATUM on UK biobanking, what did we say in our report? We noted that a national network will need to integrate a heterogeneous population of organisations and emerging networks. It's a complicated landscape that we have in the UK, large numbers of biobanks and some emerging networks, for example, the Brain Bank network. There are challenges associated with this, both in terms of management and coordination costs and overcoming barriers to interaction and communication. And we've already mentioned that in the meeting here today already. But I think it's the management costs and the coordination costs that we need to focus on too. There are also, of course, major benefits: financial savings, the potential to review and plan collections strategically across the country, across Europe. Optimized opportunities for knowledge creation, increased visibility of and access to samples, that's what we really know we want straight away, raised quality and comparability through the promotion of best practices. So those are the kinds of benefits that you get from that kind of network. So the recommendations that we came up with in relation to financing are listed here in summary. Coordination should be financed centrally by public funds, supplemented with industrial sponsorship via a fee for membership. Coordination requires dedicated funding for a UK coordination centre with permanent operational staff, with a CEO, IT lead, quality assurance lead, network development, training, ethical, legal and societal issues. There's a minimum that we need there, that you can easily think of other very important stakeholders that should be included in the network, for example, the population at large and patients in particular. Strategic management duties are really important for coordination. In other words, buying the time of stakeholders to ensure that industry or other organizations can participate in and contribute to the development of the network. And of course the activities of the coordination center, which comprise primarily travel and communication. However, there are also implications for individual biobanks. Their financing should be reconfigured. That was the recommendation. Accrual of samples should be included as costs in project proposals. That tends to be the case already, but we should review categories within those costs, such as estates and service support. Maintenance and depreciation. Central public funds should be distributed to host institutions of biobanks in the network and allocated specifically to the biobank to enable maintenance and depreciation. Access costs, we never talk about depreciation. I've never heard it discussed in any biobanking meeting, yet of course it affects us as much as anybody else, more than anybody else, because we're planning to keep our samples a long time. Therefore depreciation is a much bigger problem for us than it is for somebody who's got a nice new piece of kit from Illumina, which you know you're going to have to chuck away in 18 months anyway. So for biobanking, depreciation is a really important cost. Access costs, marginal costs should be paid to the coordination centre via grants, so when use is to be made or in a research project via grants to the coordination centre, and from fees of, for example, industrial partners. Access policy, access to be controlled via some kind of committee structure. Enrichment, that's been referred to already, different term was used, what Tim called it? The added data that comes from the new experimental annotations to a sample arising from examining the sample, doing the genotyping, whatever. And that enrichment process, returning the data to the biobank, should be mandated by funders. It should be a requirement of funding that if you are using public money or if you're participating in this network, then you've got to return the data back to where to enrich the resource so that somebody doesn't have to do the same experiment again, or so that somebody can analyze it in a different way. Okay, Stratum, yes, a final point, I think. The Stratum project also presented an exploitation plan back to Innovate UK, and this was designed to answer the question, what should we do next with the results of the Stratum project? And essentially the exploitation plan describes how to get the benefits of a national top-down policy framework, which is what this is, essentially, and simultaneously of project-oriented bottom-up development. In other words, what's going on with particular projects in particular places. How do you use the strength of a top-down structure with the strength of a bottom-up movement for a national biobanking network? And that's what we address in the exploitation plan. I don't have time to go into all that now. Okay. Conclusions. Further delay in reforming biobanking has an opportunity cost. Let's not forget that. There's economic, scientific and social costs. The economic is risk of further loss of big pharma from the United Kingdom and of falling inward investment. That was one of the clear messages that came out from our economist friends. Scientific, the failure of our country to maintain our reputation in research. This is major, 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 major problem if that's the case, and if biobanking is not seen as being an essential part of the solution of that problem. Social, relative decline ultimately in the health of the population, plus other social risks. These are major issues, really, really profound political issues, I'd go so far as to say. To help overcome institutional barriers, oh yes, right, so those are the opportunity costs. And my final point is that in moving forward, I think we need to work with innovation researchers. When I was given this work package to lead in Stratum, I didn't really have a clue what to do. Financial costs, how can I do that? Cost models, don't ask me. Was my initial reaction to Julie when she first chucked it at me. And then I thought about it. I said, okay, I'll take it on, but I'll have to do it with my innovation economist friends. And that's what I did. And I think we actually need them. Our community is too narrow. We tend to all to be scientists or representatives of patients. That's who we are. We actually need to look to the social sciences as well as the natural sciences. We need to work together to find a solution to what is ultimately a socio-economic problem as well as a technical scientific problem. And that's my conclusion.